Okay, so now we're going to move into understanding what are the organic compounds and what is their importance. So, in our body. So we have 4 major organic compounds that makes the components of our cells. So we have carbohydrates, we have lipids, proteins, and nucleic acids. So carbohydrates, they are made out of units that we call monosaccharides. Mono for one, saccharide for sugar. And they can be also made out of multiple units together that we call polysaccharides. These have a formula that it is the following. So it's CN, H2N, and ON. When it comes to some proportions, basically, or ratios, for every carbon, for every carbon you have 2 hydrogens and you have 1 nitrogen. So you can have then, for instance, C6H2 and O6, or C12H4 and O12. So the proportion is always there, or the ratio. Now for lipids, we have long carbon polymers with hydrogens on the sides. And these are sometimes as long as 40, 60 carbons with many hydrogens. And these carbons are joined to each other with the sides filled with these hydrogens. Now we have these molecules that are hydrophobic. So they are non-polar. They don't interact with water. And they include fats, oils, and steroids. Fats, for the most part, they are solid at room temperature. Oils, for the most part, they are liquid at room temperature. Proteins are made by chains of amino acids. We have 20 different amino acids. They can combine in different order, and then they can form these proteins. Proteins are what makes the majority of the components of our body, and they have different roles, including forming cells like muscle cells, the skin cells, and also they have functional properties. Like, for instance, they make enzymes. Enzymes are the proteins that helps for the chemical reactions to happen much faster, if you remember. And then nucleic acids, we have deoxyribonucleic acid, or DNA, and we have ribonucleic acid, or RNA. And they have monomers that we call nucleotides that are formed by a nitrogenous base of different types. We have 4. A sugar, that it is a sugar made out of 5 carbons. In this case, we call it pentose. And then phosphate groups attached to this sugar. And function of these nucleic acids is to store and transmit genetic information so that the cells can have a particular function, or you can replace the cells with the same structure as before, during mitosis, for instance. So here is the example of the 2 most common types of sugars that we have in our body, carbohydrates, pentose and hexoses. So a pentose is a 5 carbon ring that, in this case, this is deoxyribose. This is actually the sugar that it is found in DNA. So this is the formula, C5H10O4. You can count the number of molecules in here if you like, but something that is different between this molecule here that is called ribose, that is part of the RNA, and this is that this is called deoxyribose because it lacks an oxygen in here, while this ribose sugar has this oxygen in here. So then what you will expect is in this formula that you have an extra oxygen. So these are the two sugars that are found in nucleic acids. This one for DNA and this one for RNA. And it is in this end, the 5' end that they call 5' end, where you have attachment of the next nucleotide. And then you start elongating this DNA or the RNA. And then here you have the 3' end, where you also can attach these molecules. So here we have hexoses, 6 carbon sugars. So this is the formula, C6H12O6. I don't know what I said at the beginning with these carbohydrates, but basically for every carbon and oxygen, you basically have the double amount of hydrogens. So in this case, this is the formula, C6H12O6. And look at the formula here for fructose and galactose. Are they the same? No. The three of them are carbohydrates, but they have different configuration. They have the same number of atoms of each kind, like 6 carbons, 6 oxygen, and 12 hydrogens, but the way they arrange are different. So in the case of these two, fructose and galactose, they are called isomers of glucose because they look like glucose, but they are not the same as glucose. So this one arranged differently than this, and this one it's almost the same, as you can see. What is the difference? Look at how here we have this hydroxyl group, but not in here. And then see how in here we have this hydroxyl group, but not in here. So these are almost like mirror images. So these are isomers. So basically carbohydrates are categories that they may have the same, in this case, they might have the same number of atoms of each kind, but they look different. If someone will give you 500 grams of flour with 100 grams of sugar, and then 100 grams of butter, 100 milliliters of milk, and they ask you, make something out of it. Well, you can make a cake, you can make cookies, you can make pancakes. Well, plus the eggs, right? And they give you 2 eggs. So you can be creative. It's the same amount of ingredients that you can give to multiple people, but they come up with different products. So similarly here we have isomers, and all of these will be things that you can consume. In this case, I mean, if you don't make pancakes, but you do different things, they can be pastries, okay? So those are then the isomers. Same formula, but different structure or configuration. Now, sometimes you join these carbohydrates to store them. Or they may come like that in the items that we ingest. Like for instance, starch, it's polymers of glucose. They are all joined together. And we have basically disaccharides like lactose, which is the sugar of milk, that it has glucose and galactose, or sucrose, which is glucose and fructose. So sometimes naturally the foods that we ingest may have these disaccharides or polysaccharides. But in our body, we can store these carbohydrates or we can utilize them, and we need to absorb them. So here, for instance, we have these monomers like glucose and fructose that if you remove water, you can convert them into sucrose. We don't do that in our body, but we can do the opposite. Like if you ingest sugar, this fructose that is joined by this bond with glucose, you add water and you can break it down. So you can have glucose and fructose, which are monosaccharides that you can absorb and that you can utilize. If you find a $2 bill, you're lucky. But what about if you want to use it? I mean, cashiers, they rarely will basically, I mean, they will take it for sure for them, and then they give you to split dollars or coins if you want out of their pocket. But basically in this case, I mean, you will have to split or get a new bill, right? 2 bills of $1 to spend it. So similarly, these molecules, these saccharides, you have to break them in order for you to ingest, okay? So we have enzymes in our intestines that do that. They can break this bond, and then you can have these 2 molecules. And now, this is glycogen. These are multiple glucose molecules joined and branching out of different parts. And this is like our storage form of glucose. Whenever we eat food, depending on what you eat, you may have a meal rich in carbohydrates or balanced, like you may have appropriate amount of proteins, carbohydrates, and also lipids. But something that our body does, it is use whatever it needs first, and then the second thing that it's going to do is to store the excess. The first storage form that we will have, or we will make, will be glycogen. Our liver will store glucose. And then also our muscles, and also our heart. Well, our skeletal muscles and our heart, they can store glycogen. Now, something that what will happen with the glycogen that we have in the liver, just in case that you're fasting and your glucose levels drop, then the glycogen will be broken down by an enzyme into what we call glycolysis. We'll see that later on. Glycogenolysis. And then you will have your blood glucose levels going up again for about 2 hours. Now, our muscles in our skeletal system, they won't give the glycogen to increase your blood glucose levels. They will keep it because they need to contract. So they need to have their own source of energy. If you're in a relationship and that person gives you 100% of their check, oh my gosh, you're lucky. Yes. But typically, people give a certain percent in a relationship of their check to the other person and keep something for their own expenses. And also don't give the 100%. But don't give too little as well, if you need to give. Okay, so we already saw nucleic acids, we saw carbohydrates, now let's look at lipids. So again, lipids are linear chains of carbons with a lot of hydrogens in it. So because you have all this hydrogen in them, these type of molecules, they don't have a charge. You saturate all of these bonds that you can possibly have with atoms that of hydrogen, the carbons that you don't end up with a charge. Now, here at the end of this molecule, we have a carboxyl group, which is this one in here. And this formula represents this model, which is palmitic acid. So this is what we call a saturated fatty acid. We will see what is saturated versus unsaturated. Now, this molecule, which is similar to this, it has a band in here. So all of these are single covalent bonds, and here you have a double covalent bond. When you have double covalent bonds, you can create a little band in your molecule. In this case, this is oleic acid. And this structure, since it has this band, is less rigid, and it will not clog your arteries. So this is what we call monounsaturated, because you have only only one double covalent bond. Now, the more covalent bonds that you have, the more bending or kinks that you will have in the molecule. In this case, you have 2 double covalent bonds, but you will have more. So this is what we call polyunsaturated, and this is what we call linoleic acid. This is 2 types of molecules that are not bad for our body, while this one, it is. So that's saturated versus unsaturated. Now, we have also triglycerides, which are 3 fatty acids. So these are short chains of carbons that are bonded to one glycerol molecule. So tri for 3 fatty acids and glycerides for glycerol. So this is how glycerol looks like. It's 3 carbons, and then they have a hydrogen on one side, and at the ends also have hydrogen bond to them, and then they have hydroxyl groups. So when you dehydrate this molecule by extracting this hydroxyl group from these 3 fatty acids, and then hydrogen from this glycerol, you will end up dehydrating these molecules by joining them. So you form a triglyceride. And then you can do the opposite. You can break the bonds by adding waters, and you end up with 3 fatty acids and glycerol. So lipids can also be stored. After someone eats a meal, it uses as much glucose from that meal as it was necessary, and then it stores glycogen in the liver and also in the muscle cells and in the heart. The excess of glucose that you will have, you will convert it into lipids. And these lipids will go into the adipose tissue, the fat tissue that we have in the body, under the skin, the subcutaneous tissue, and other areas of the body. And then these ones you will use for later. Okay, so it's a storage energy, basically, triglycerides. Another type of lipids that we have are the phospholipids. Phospholipids, they have 2 fatty acid tails bonded to a phosphate group. And then you have a glycerol in between the fatty acids and the phosphate group. So they form like a bridge. And in here, in this phosphate group, you have polarity, you have a charge. So this charge can interact with water. So this part is hydrophobic, while this one is hydrophilic. Phosphate groups have a negative charge. So these can interact with the positive charges from water, but these ones can't. So these phospholipids form part of the plasma membrane in our cells, which is basically a wrapping around our cells that makes our cells to be separate units. It's like these walls in this room or in your house. You have a separate unit. Your house is independent from your neighbors' houses. So these basically faces internally these molecules of phospholipids, while this one faces externally and also internally. The other type of lipids that we have are molecules with 4 rings that we call steroids. They are derived from cholesterol. Cholesterol, it's a molecule that is naturally found in certain foods, especially animal foods, but also it is produced by our body. Certain people, they have defects in the receptors for the cholesterol molecules, and then they end up accumulating a lot of cholesterol in their blood. And they have to decrease the intake of animal products to decrease the levels of cholesterol, but somehow they still have it high, especially if they don't take these drugs that decreases the levels of cholesterol. Why is this? Well, because our liver still produces the cholesterol. But why do we need cholesterol? Well, because cholesterol, we convert it into sex hormones, and also it is part of the plasma membrane. This wrapping that I told you around the cells is needed in there. So all that it changes in the sex hormones that we have is all of these side groups, but they have these 4 rings attached to each other. Questions so far about lipids, carbohydrates, or proteins? Sorry, not proteins. Lipids, nucleic acids, or carbohydrates? Okay, now let's talk about proteins. So proteins are made out of amino acids. Amino acids are made of amino groups, like this nitrogen with these 2 hydrogens, a central carbon that always have a hydrogen attached to it, and it has an R group. What is this R group? Well, an R group, it is a structure that varies in their configuration, and it gives specific properties to the amino acids, making them different. We have 20 different amino acids. Out of these 20 different amino acids, 11 are non-essential and 9 are essential. Does it mean like we don't need them all 20? No. What it means is like the non-essential, we cannot make them. But the 11, we can make them by changing this R group. So here are the examples of amino acids. The simplest amino acid that we have, it is glycine. It has this amino group, it has this central carbon, it has this hydrogen. Oh, and I forgot to tell you, amino acids also have this carboxyl group, which basically it is this carbon with these 2 oxygens and this hydrogen. So this is the core. All amino acids has this core, and they vary just based on different R groups that they have. So then, if you see this hydrogen, this is the R group for glycine. And then tryptophan, it has these rings and this methyl group that attaches this two ring to this core. So this is a non-polar amino acid. This one doesn't have a charge, tryptophan. And then this molecule, or this amino acid, is called cysteine. It is a polar amino acid, so this R group, it is charged and allows it to interact with water, for instance. So in here we have this group that is called sulfahydryl. So it's a sulfur with a hydrogen, sulfahydryl group. Or it's a group. So all are the same in the core, but differences here in the R group is what makes them to be a specific amino acids. So some amino acids, they can have a negative charge, and some amino acids, they can have a positive charge, and some of them, they don't have a charge at all. So the ones that has a positive charge are basic amino acids, and the ones that don't have a charge is neutral amino acids. So in order to make a protein, what you will do is dehydrate these peptides when they are together. You dehydrate them and then you join them by a bond that we call peptide bond. So in here we have alanine, another type of amino acid, and then we have glycine. So you take away from this carboxyl group from alanine, you take away from this, carboxyl group, you take away this hydroxyl group, OH group, and then from this glycine, from the amino part, which is the NH2, you take away the hydrogen. So then here you end up with water, you dehydrate it, and then you form this dipeptide bond. So then if you want to break it, you basically add water, and then this is called hydrolysis. You attach one hydrogen to glycine, and then the hydroxyl group to alanine, or to this course of peptides. So you can basically make a bond and form dipeptides, or break a bond and form single amino acids. So through these peptide bonds at many, if you have many amino acids, again we have 20 different amino acids, they are encoded by the DNA. When you're coding for proteins in your DNA, you have 3 nucleotides that can tell specifically what amino acid you need. So it's like a code. And then that DNA will be made into messenger RNA, and this messenger RNA is going to exit the nucleus of the cell where DNA is found, and then it's going to be read by the ribosomes, which are little structures in the cytoplasm of the cells that can read that code, and they can bring these specific amino acids to make the protein. So basically, you read groups of 3 nucleotides to make a single amino acid, and some of these, they have variation in the code because at the last nucleotide, you can have in some amino acids a different one that codes for the same amino acid. So it's a little bit different code, but it means the same. Back in the day, when a woman wanted to get attention of a gentleman, he dropped a handkerchief, and the guy picked it up. And then they smiled, and they started a conversation. It was a code of liking one another, right? Nowadays, what you do, you text, same deal, right? It's different way of saying the same thing. So we have different codes. Same message. So here is what we call the primary structure. So when you join a specific, or when you join the amino acids by a peptide bond, you start forming a chain of amino acids. This is going to be the primary structure or a polypeptide. You have single amino acids, you join them together, you form dipeptides, you form tripeptides, tetrapeptides, you keep on going like that. After 20 amino acids, you call it polypeptide. And then they are joined by these peptide bonds. The carboxyl group joined to the amino group. So in here you have glycine, serine, cysteine. And then you can add alanine, tryptophan, tyrosine, any of the 20, and then you form your protein. So then the unique linear sequence of amino acids is what is the primary structure, and this is determined by DNA. So this linear chain of amino acids, the polypeptides, can form helixes like this, or they can form a pleat. This is called the secondary structure. So this is an alpha helix, and this is a beta pleat. And what it makes them to fold that way? Well, in this case will be hydrogen bonds. So primary structure is formed by peptide bonds. Secondary structure is formed by hydrogen bonds. And now this secondary structure can fold again and form the tertiary structure. And this folding, it gives a particular shape to the proteins. That shape is important. Whether this will be a part of a larger unit or a larger protein, or whether it's just the final shape. Without that specific shape, the proteins, they don't work, especially if it's enzymes. And when you lose that shape, for whatever reason, higher temperature, high acidity, high concentration of hydrogen ions, you call that process denaturation. The proteins in the eggs that you cook, for instance, in a pan in the morning or in the afternoon, whatever it's your likeness. So you break the egg, you put it in the pan, and then that change in the white from clear color to white opaque color is the denaturation process. So that's what happened with proteins. In this case, this secondary structure folds. It forms a globular protein, a protein that it looks like a sphere, made by alpha helices and beta pleats. So what makes them to fold like that? Well, interactions between the charges of these beta pleats and the alpha helices. Or maybe the lack of interaction. The lack of interaction sometimes can leave gaps in the proteins, like in the case of these enzymes, so that you will have a gap to feed a substrate so that you can bring molecules together and join them. Or maybe break the bonds apart, depending on the enzyme that you have, whether it's catabolic to break down a bond or anabolic to make a bond, to join smaller molecules to make it larger. And then we have fibrous proteins in our body that form filaments. So these shapes are stabilized by hydrogen bonds, ionic bonds, and hydrophobic interactions. So it's not only making the shape, but also stabilizing it. Because we will have changes in body temperature, changes in pH. As long as they are not extreme, you can still have functioning proteins. That's the point. And that's why we have these bonds that help them to maintain that particular shape. Now, some proteins can have what we call a quaternary structure. So in the quaternary structure, you can have globular proteins joining together, like hemoglobin, that is made out of 4 units, or fibrous proteins can form helices, like collagen. Collagen, the abundant protein in our skin, is made out of 3 filaments, 3 fibrous proteins that forms a helix, a triple helix. That's the final 3-dimensional shape of these proteins. And they have that shape for a particular reason. In the case of hemoglobin, it will be to carry oxygen and deliver it to the cells and pick up CO2 and deliver it to our lungs so we can exhale the CO2. In the case of these fibrous proteins, to create resilience. So you can basically just try to stretch the skin, but it will go back, it will recoil to its normal shape. Keratin gives that particular structure to our hair. More details about nucleic acids. So, nucleic acids are made of a pentose sugar. If it's RNA, it will be a ribose. If it's DNA, it will be deoxyribose sugar. And remember what I told you, I think I pointed this one as, this is a 3', this is a 5'. So basically, you will join a nitrogenous base to this pentose sugar and a phosphate group here, and then you will have a nucleotide. And this nitrogenous base varies in name also as in the number of rings. So these double ring structures, when one of these is attached to the pentose sugar and then to this phosphate group, you will call it differently. So for instance, adenine, when it binds into here, and then you have only one phosphate group, you will be calling it adenosine monophosphate. If you have 2 phosphate groups, you call it adenosine diphosphate. If you have 3 groups, you will call it adenosine triphosphate. Now, that's not a nucleic acid, that is a energy molecule. It's like the currency of energy from our body, ATP. But it is derived from this adenine. Now, these nitrogenous bases that has double ring are called purines. The ones that only has 1 ring, it will be called pyrimidines. So the one with the shorter name has the longer structure, the one with the longer name has the shorter structure. And in this case, this one is called adenine, and this one is called guanine. So then you will have one of these attaching to here, and then you can have another one, and then you will have your bases, DNA bases, RNA bases. Now we have here these pyrimidines. So we have cytosine, thymine, and uracil. Uracil is only found in RNA, but not in DNA. And then they base pair on opposite sides of the DNA. The DNA, basically, it is two strands. In the case of the human cells, it has two strands that base pair in the middle. So here is the molecule of ATP all together with these different components. So if you only have the ribose with this nitrogenous base, you call it adenosine. When you add one phosphate group, it's called AMP. When you add two phosphate groups, it will be ADP. When you have three, will be ATP. And the chemical bonds between these molecules drives energy when they're broken. So basically, from ATP to ADP, you release energy. Now, if you want to make ATP, you have to add a phosphate group into this ADP, and then you will form it again. So this is the DNA structure. It's made out of two long chains of these bases. So you join them from 5' to 3', and you form these single-stranded molecules first. This is single-stranded RNA, we'll look into that, but basically it's two strands. So you have here one strand of DNA, and then here you have another strand of DNA, and they run in opposite directions. This is like, let's say this is the 5' end, and this is the 3' end of this strand, and this will be the 3' end, sorry, the 5' end of this strand, and the 3' end of this strand. So they run into what we call antiparallel, and these nitrogenous bases face medially or to the middle, while the sugar backbone, the ribose sugar, is found externally. And then you always base pair in DNA cytosine with guanines and adenine with thymine. So if you magnify one of these, you can see how they base pair. In this case, we have a guanine, and then on this other strand, you have a cytosine. So you have here these hydrogen bonds running between this purine and this pyrimidine. So you have between guanine and cytosine 1, 2, 3 hydrogen bonds. And then here between adenine and thymine, you have 2 hydrogen bonds only. So you always base pair G's with C's and A's with T's. They form pairs. Based on the fact that G's and C's, they have 3 hydrogen bonds, and that A's and T's has 2 hydrogen bonds, which bond would you say is most likely to be broken? A and T or G and C? A and T, because you only have 2 hydrogen bonds. So G's and C's, they have triple bond, so it's harder to break down. It's like a rope. You can have a rope with 2 strands, or you can have with triple strand, and the strongest will be the one with 3 strands. Okay? And then there could be regions of DNA where you have multiple G's and C's and not so many A's and T's. So that region will be strongly together than other regions where you have more A's and T's than G's and C's. And then we have a code then depending on the sequence of the bases of DNA that can make us to form an RNA with a specific bases. So one thing for sure is that instead of T's in your RNA, you have uracil. But you don't have thymines. Any questions? Okay, so in the next chapter, we're going to see the structures of the cell, the functions of these structures, and also how the cells transport molecules, how they communicate, and how they divide. Test your knowledge questions. Which type of bond involves unequal sharing of electrons between the atoms? Polar covalent, ionic, nonpolar covalent, or hydrogen bonds? Polar covalent. Very good. What does molarity measure? Number of particles per kilogram, number of moles of solid per liter of solution, number of atoms in an element, or number of electrons in valence shell? Number of moles of solid per liter of solution. True or false question? Oxygen gas is a molecule but not a compound, while water is both a molecule and a compound. Why?
Because the molecule is 2 atoms and a compound is 2 or more different atoms.
The clue is, or the key is difference. Okay? Now, for this next question, ionic bonds formed by transfer of electrons between atoms, creating charged ions that attract each other. True or false? It's a long question, right? So when you transfer electrons from one molecule to another, what type of bond it is? When you share is covalent, but when you transfer it is what type of bond? Hydrogen bond, ionic bond? And then what happened when you have transfer of electrons to one molecule and the other one accepted? What charge does electrons have? Negative. So when one molecule accepts an electron, becomes negatively charged, right? And the other one that donates will be positively charged. How do you call those? Ions. When you donate, you become positive. Now, if someone gives you something, if you accept a gift, that doesn't make you negative, but in the case of molecules, it is. So then ionic bonds formed by transfer of electrons between atoms, creating ions that attract each other. True or false? You don't say it loud.
True.
True. Normal blood osmolarity ranges between 275 and 295 milliosmoles per liter, which help regulate water balance and maintain homeostasis in the body. True or false? Not true nor false. Okay. Describe the 4 levels of protein. I didn't say the answer because you didn't say it, okay? But you will answer yourself. So describe the 4 levels of protein structure and explain why tertiary structure is essential for protein function. What are the 4 levels of protein structure first? You just saw it?
Is it the primary, secondary, tertiary, quaternary?
Yes. So it's from primary to quaternary. And now, why tertiary structure is essential for protein function? Sometimes you receive products through the mail that you order from a company, and they don't come assembled. They don't have that tertiary structure. So what it is needed for that device to function? It needs to have the tertiary structure, right? That particular shape. It's like it sometimes happens. I mean, what if you get like a vacuum cleaner, chop back, most likely. The other ones are, for the most part, almost assembled, like the ones for carpet. But what if you get a chop back that basically the lid is not on, and then it needs the attachment of the hoses to where it has a vacuum, and then put the little wheels into it? I mean, if you don't do that, and you're trying to vacuum something, is it going to work? No. So similarly, our proteins, they have to have that three-dimensional shape to function. Otherwise, they don't. So that's the answer. Okay, so